Regulating the Cell Cycle, Chapter 11, Section 3. Learning objectives: investigate how the cell cycle is regulated, compare cancer cells with other cells, investigate how cells become specialized for different functions, explain what stem cells are, and evaluate some possible benefits and issues associated with stem cell research. How do cells know when it's time to divide? One striking fact about cells in multicellular organisms is how carefully cell growth and cell divisions are controlled. Careful control of the cell cycle is essential for orderly growth and development. If something goes wrong with the control, serious diseases such as cancer sometimes result. When scientists grow cells in the laboratory, most cells will divide until they come in contact with one another. Once they do, they usually stop dividing and growing. Something similar happens inside the body. When an injury such as a cut in the skin or a break in the bone occurs, cells at the edges of the injury are stimulated to divide rapidly. New cells form, starting the process of healing. When the healing process nears completion, the rate of cell division slows. Controls on growth are restored and normal activities return. Scientists have identified dozens of proteins that help to regulate the cell cycle. The cell cycle is controlled by regulatory proteins both inside and outside the cell. One group of internal regulatory proteins responds to events inside the cell. These proteins act as checkpoints, allowing the cell cycle to proceed only when certain events have taken place. Proteins that respond to events outside the cell are called external regulatory proteins. External regulatory proteins direct cells to speed up or slow down the cell cycle. One important group of external regulatory proteins is a group made up of growth factors. Growth factors stimulate the growth and division of cells. These proteins are especially important during embryonic development and wound healing. Biologists have been discovering for years a signal that regulates the cell cycle because they realized that it could help them treat diseases. Learning that there is not just one signal but many has made the job more complicated. Scientists discovered the first regulatory protein in the early 1980s. When they injected the protein into a non-dividing cell, a mitotic spindle would form. They named the protein cyclin because it seems to regulate the cell cycle. Investigators have since discovered a family of proteins known as cyclins that regulate the timing of the cell cycle in eukaryotic cells. Cyclins rise and fall in a regular pattern. Just as new cells are produced every day in a multicellular organism, many other cells die. A cell may die by accident due to the damage or injury, or a cell may actually be programmed to die. Apoptosis is the process of programming cell death. Once apoptosis is triggered, a cell undergoes a series of controlled steps leading to its self-destruction. First, the cell and its chromatids shrink, and then parts of the cell membranes break off. Neighboring cells then quickly clean up the cell's remains. Why is cell growth regulated so carefully? The principal reason may be the consequences of uncontrolled cell growth in multicellular organisms. Cancer is a disorder in which body cells lose the ability to control cell growth. Cancer cells do not respond to the signals that regulate the growth of most cells. As a result, the cell cycle is disrupted, and cells grow and divide uncontrollably. Cancer cells form a mass of cells called a tumor. However, not all tumors are cancerous. Some are benign or non-cancerous. As cancer cells spread to the surrounding healthy tissue, they absorb the nutrients needed by other cells, block nerve connections, and prevent the organs they invade from functioning properly. Soon, the delicate balances that exist in the body are disrupted, and life-threatening illnesses result. Cancers are caused by defects in the genes that regulate cell growth and division. There are several sources of such defects, including smoke or chewing tobacco, radiation exposure, and even viral infection. All cancers, however, have one thing in common. The control over the cell cycle has broken down. Some cancer cells will no longer respond to external growth regulators, while others fail to produce the internal regulators that ensure orderly growth. An astonishing number of cancer cells have a defect in a gene for a checkpoint protein known as p53, which normally halts the cell cycle until all chromosomes have been properly replicated. Damaged or defective p53 genes cause cells to lose the information needed to respond to signals that normally control their growth. When a cancerous tumor is localized, it can be often removed by surgery. Skin cancer, one of the most common forms of the disease, can usually be treated this way. Melanomas, the most serious form of skin cancer, can be removed surgically, but only if spotted very early. Cancerous tumors tend to grow more rapidly than normal cells. For this reason, they need to copy their DNA relatively quickly. This makes them especially vulnerable to damage from high-energy radiation. As a result, many tumors can be effectively treated with careful targeted beams of radiation. Medical researchers have worked for years to develop chemical compounds that would kill cancer cells as well, or at least slow their growth. The use of such compounds against cancer is known as chemotherapy. Great advances in chemotherapy have taken place in recent years and have made it possible to cure some forms of cancer. However, because most chemotherapy compounds target rapidly dividing cells, they also interfere with cell division in normal healthy cells. This produces serious side effects in many patients. Cancer is a serious disease. Understanding and combating cancer remains a major scientific challenge, but scientists at least know where to start. Cancer is a disease of the cell cycle, and conquering cancer will require an even deeper understanding of the processes that control cell division. Animals and vascular plants pass through a developmental stage called an embryo, from which the adult organism is gradually produced. During the developmental process, cells become more and more different from one another and become specialized for particular functions. The process by which cells become specialized is known as differentiation. During development of an organism, cells differentiate into many distinct cell types. A differentiated cell has become quite literally different from the embryonic cell that produced it and specialized to perform certain tasks such as contraction, photosynthesis, or protection. Our bodies and all the bodies of all multicellular organisms contain highly differentiated cells that carry out the jobs we need to perform to stay alive. One of the most important questions in biology is how all of the specialized differentiated cell types in the body are formed from just a single cell, the fertilized egg called a zygote. Biologists say that the zygote is totipotent, literally able to do everything to develop into any type of cell in the body, including the cells that make up the extra-embryonic membranes and placenta. Only the fertilized egg and the cells produced by the first few cell divisions of the embryonic development are truly totipotent. If there is a secret by which the cells start the process of differentiation, these are the cells that know that secret. After about 4 days of development, a human embryo forms into a blastocyst, a hollow ball of cells with a cluster of cells inside known as inner cell mass. Even at the early stage, the cell of the blastocyst has begun to specialize. The outer cell form tissues that will attach to the embryo to its mother, while the inner cell mass becomes the embryo itself. The cells of the inner cell mass are said to be pluripotent. Pluripotent cells can develop into any cells in the body, although they generally cannot form the tissues surrounding the embryo. As the name implies, stem cells sit at the base of the branching stem of a development from which different cell types form. Stem cells are the unspecialized cells from which differentiated cells develop. Stem cells are found in the early embryo, of course, but they are also found in many places in the adult body. Pluripotent embryonic stem cells are more versatile than adult stem cells, since they are capable of producing every cell type in the body. Cells in some tissues, like blood and skin, have a limited lifespan and must be constantly replaced. Pools of adult stem cells found in various locations throughout the body produce the new cells needed for these tissues. New blood cells differentiate from stem cells found in the bone marrow, and many skin cells are found in hair follicles. Small clusters of adult stem cells are even found in the brain, in the heart, and in the skeletal muscle. These adult cells are referred to as multipotent. Because the types of differentiated cells they can form are usually limited to replace cells in the tissues where they are found, they are called multipotent. Basic research on stem cells takes on a special urgency in the light of the importance it might have for human health. Heart attacks destroy cells in the heart muscle, strokes injure brain cells, and spinal cord injuries cause paralysis by breaking connections between nerve cells. Not surprisingly, the prospect of using stem cells to repair such cellular damage has excited medical researchers. Stem cell harvests from the bone marrow might be cultured and then injected into damaged portions of the heart. Once in place, the stem cells would learn what kind of cells they needed to be from the surviving cells around them. The stem cells would differentiate to become new heart muscles cells. Because adult stem cells can be harvested from a willing donor, research with these cells has raised few ethical questions. This is not the case with embryonic stem cells, which are generally obtained in ways that cause the destruction of an embryo. For this reason, individuals who seek to protect human embryonic life oppose such research as unethical. Other groups support such research as essential for saving human lives, and argue that it would be unethical to restrict research. Human embryonic stem cell research is controversial because the argument for it is and against it both involve ethical issues of life and death. A fundamental breakthrough took place in 2007 in Japan, where a doctor was able to convert human fibroblasts into cells that closely resemble embryonic stem cells. His work is summarized in the picture on this page. These induced pluripotent stem cells, or iPS cells as they are known, are now widely used in research. Under certain conditions, iPS cells may be able to replace embryonic stem cells.